Plant pathogens are harmful microbial agents that can cause infections. Recent research underscores the severity of this issue, revealing that plant diseases are responsible for up to 16% of potential production losses worldwide. Our review focuses on Fusarium wilt. It is a disease that affects tomato crops globally. The immunity I gene is a key player in unraveling the complexities of plant defense mechanisms against these deadly pathogens. To understand how plants defend themselves, we delve into their strategies. Plants employ two main defense mechanisms. Firstly, pattern recognition receptor (PRR) on the outer surface of cells detect pathogen-associated molecular patterns (PAMPs). Secondly, the effectors, which are proteins secreted by the pathogens, trigger effector-triggered immunity (ETI). So, these defense strategies play a pivotal role in determining the success or failure of the entire harvest. In this review, we investigate the defense mechanism employed by tomato plants against the Fusarium wilt. We also investigate the potential of essential oils from aromatic plants as alternative biofungicides when applied with biocontrol agents.
Soil-borne fungal pathogens known as Fusarium oxysporum has colonized in usually in vascular tissue of tomato, resulting in a deadly illness that causes wilting and also tomato death. So the pathogen Fusarium oxysporum liposicai, which we call it as FOL, in tomato plants contains a variety of virus genes that we call it as AVR1, AVR2, and also AVR3. So the gene for gene theory described this resistance by explaining the relationship between the pathogen races, which is the virus genes, and also the host plant cultivars through the interaction of this AVR gene and also the resistant gene, especially we call it as an immunity gene. According to recent research, FOL causes the tomato vascular wilt and reduce the yield to the maximum extent as possible. So due to the significance of for agriculture, tomato wilt disease caused by this FOL has been well investigated and also continues to be one of the most well-studied plant pathogen interaction involving a vascular wilt pathogen.
So moving on to the plant defense mechanism. Plant defense system include PTI and ETI. PTI as the initial defense to quickly respond to common pathogen patterns. So the mechanism in tomato first is the recognition of the PAMP by the PRR receptor designed to recognize specific PAMP, which commonly includes chitin, beta-glucans, and other cell wall components. When PRR detect this signal, they will undergo activation, setting off intracellular signaling cascades such as the MAPK pathway. The activation of MAPK pathway will result in the phosphorylation and the activation of transcription factor, which in this case is WRKY. It will involve in the synthesis of the defense-related protein and antimicrobial compound. One of the antimicrobial compound is reactive oxygen species (ROS), which being produced by NADPH oxidase. So this process is important in oxidative burst where it can damage pathogen structures and hindering their growth as well as signaling the activation of phytohormones such as salicylic acid. Other compound is callose, which it will be deposited to the cell wall, forming a barrier at the infection site and creating a robust physical impediment and restrict pathogen ability to penetrate and spread to the plant tissue. So the deposition of callose and ROS are integrated in the reinforcement of the cell wall, providing a coordinated and comprehensive response to protect the tomato from wilt disease. PTI will also activate calcium ion influx, which act as a second messenger to elevate cytosolic calcium level and activate Murray signaling pathway involved in plant defense system. So calcium ion also participate in oxidative burst, cell wall reinforcement, regulate the expression of defense-related gene as well as the induction of the programs of death in the infected cells such as hypersensitive response, which limits the spread of the pathogens. So, the activation of the salicylic acid pathway is crucial in the induction of the systemic acquired resistance, a long-lasting defense response that enhances the plant's ability to resist subsequent infection. So in tomato, SA signaling also leads to the expression of a set of defense-related genes involved in SAR, such as PR1 genes encodes PR1 protein, PR2 genes encode glucanase, and PR3 genes encodes chitinase. These genes will contribute to the plant's defense mechanism. So that's all for PTI mechanism in tomato wilt disease.
The another immune system is effector trigger immunity (ETI), which is activated by detecting the effector that directly delivered by the pathogen. The effectors are proteins that manipulate host process and establish infection. In the case of tomato plant 2 against 4, resistant R protein will recognize the effector and undergo conformational change. These activated R proteins initiate and activate downstream signaling pathway including MAPK pathway and thus lead to transcriptional activation of defense-related genes, and this will cause the synthesis of antimicrobial protein secondary metabolite and phytohormones. The distinct feature of ETI is hypersensitive response (HR) that is rapid and localized self-death as infection site. It limits pathogen growth and spread and associated with ROS production. This will cause the oxidative damage on lipid, protein, and DNA that will change the membrane characteristics such as fluidity. ETI result in a stronger immune response and alters specific pathogen effectors. It will cause the development of long-lasting immunity, including systemic acquired resistance that activate by the salicylic acid and induced systemic resistance that activate by jasmonic acid. In the research done in 2013, they mentioned that this pathway will suppress the disease by 20 to 85%. In conclusion, for tomato plant 2 against 4, ETI work in concern with PTI to provide a more comprehensive defense strategy.
Okay, so moving on to the immunity gene. So resistance genes, as I mentioned before, which is, for example, like immunity gene that contains of I2, and also I3 in tomato cultivar. So it were investigated to analyze its resistance against the four respectively. So the presence of introns in the I gene suggests that it has a possibility of for alternative splicing, which will allow the synthesis of various protein isoforms with different function, especially the I protein. So the I protein, also known as a somatic embryogenesis receptor kinase 3 brasinosteroid associated kinase SERK3-BAK1, is a receptor-like kinase that play important role in a plant innate immunity. So the structural properties of the I protein are important to its function, which the I protein structural properties such as the presence of the kinase domain are essential for its protective role in plants. So let's move on to the figure that I put. So which explaining about the interaction between the immunity gene and also the virulence stress gene that crucial for full resistance. So if you can see in the photo, A and B shows that the diverse phenotypes of F1 plants, or specifically tomato cultivar, resulting from crosses between two types of cultivar, which is GCR161 and also C32, shows that the presence of the virulence stress genes give influence different phenotypes between each other. So moving on to the C. So C shows that the F2 progeny with a presence of a virus 1 transgenic hybrid give a different phenotypes too. You can see the first sample in the photo on C shows that the cultivar is dying, which shows that the sample had a severe attack by the virus 1 gene. So moving on to the number 2, we can see that the cultivar stunted with necrosis, which show a significant growth impairment accompanied by tissue death. So the third one is we can see a curling leaf, which displaying a growth retardation along with a characteristic leaf curling. So the last one is the number 4 sample. We can see it has a very healthy growth, which reflecting unaffected by the virulence genes.
The zero weight is a prevalent disease in tomato productions. Integrated management of zero weight in tomatoes is crucial to minimize the reliance on synthetic fungicides. By implementing these alternative methods, the impact on the environment is lessened and fungus populations are kept from developing resistance. This can be managed using essential oil as an alternative biofungicide. These oils have antifungal properties, reducing disease severity and enhancing tomato plant growth. Induced whole resistance is another method used to control fusarium, activating short-term signaling pathway related to tomato plant defense. This study evaluated the potential of citronella and melaleuca essential oil in managing fungus fall, revealing their potential in enhancing callus deposition and reactive oxygen species production on seed surfaces. The long-term effects of this application with minimal essential oil requirements have opened new opportunities for developing biopesticides based on essential oil in agriculture. However, this method is cost ineffective as essential oils are often expensive due to the labor-intensive processes involved in their production. It takes a large amount of plant material to produce a small amount of essential oil, and the extraction methods can be time-consuming. Another study here shows the soil drainage application with chemical resistance inducers such as gibberellic acid, sorbic acid, cytokinin, and auxin also show greater changes than foliar spray in shoot length, root length, fresh weed, and dry weed.
In conclusion, fusarium wilt that is caused by fall is a significant threat to tomato plants. In response to this disease, tomato plants employ various defense mechanisms. These include pattern recognition receptor triggering, which initiate PAMP-triggered immunity, and the subsequent effector-triggered immunity, involving the activation of specific gene, such as the enigmatic I gene. To effectively manage the fusarium wilt and overall improvement on tomato plant health, a combination of essential oil and biocontrol agents has been found to be effective. Resistant inducers such as diterpenoid play a crucial role by targeting the root system where the infection typically begins. This enhances the plant's resistance to fusarium wilt, contributing to a more robust defense against the disease.